we were discussing about the origin of different cells, okay? Particularly the lymphocytes which come out of the lymphocyte progenitor cells in the bone marrow. And I told you that the lymphocyte progenitor cell is going to give rise to two different types of cells. One type of cells are the small lymphocytes, and the second type are the large lymphocyte cells. The large lymphocyte cells are granular. There are granules in its cytoplasm which are filled with different cytotoxic enzymes. And we call these cells as the natural killer cells. And the natural killer cells, you know that we call them natural killer cells because they naturally are equipped with this capability that they can kill cells that are infected with viruses. Some of the pathogens, they live inside the cells. Some of the pathogens, they live outside the cells. Most of the bacteria, for instance, would live outside the cells. But the viruses mostly are found inside the cells. It's very difficult for different components of the immune system to recognize these viral particles because they are not mostly out there in the tissues or blood. They are intracellular. So that's why the immune system has evolved some features in order to identify cells that are infected with viruses. Okay, so natural killer cells are one of them. They also secrete different kind of cytokines that impede replication of these viruses. Not only kill the cells directly, but they can also, do secretion of different cytokines, which are peptides secreted by these cells, which can impede the replication of viruses. The small lymphocytes are cells of the adaptive immune system, which also come out of the lymphoid progenitor cells, and they are small because they are functionally inactive. When they are formed in the bone marrow, we are talking now about the B cells and the T cells. When these cells are made in the bone marrow, they are smaller in sizes because they are not active yet. They would be activated once these cells would come in contact with some kind of pathogen, and that can happen only in some tissues that we are going to discuss here, the lymphatic system. We would also discuss in detail where these cells are going to come in contact with the pathogen for the first time. So these, when come in contact with the pathogens, would turn into effector cells. Effector cells are functionally active cells doing some kind of a job. For instance, the B cells, when they become effector cells, these cells would start secreting different kind of antibodies against the pathogens, very specific for the pathogens. And the T cells have various effector functions. Some are cytotoxic T cells, which are going to kill the virally infected cell directly, just like the natural killer cells, okay? And some of these cells are going to help other cells with their functions. For instance, the helper T cells can help with the functions of the B lymphocytes. They can, stimulate the B lymphocytes to turn into memory cells or plasma cells. The memory cells are going to keep that memory of the pathogen and reside there in the body for longer periods of time. And the plasma cells are going to secrete antibodies, Then there are regulatory T cells, there are memory T cells. We are not going into detail of these cells. Now, if you see, the B cells, they are equipped with a B cell receptor, and the T cells are equipped with, a T cell receptor. How the cells of the adaptive immune system are different from the cells of the innate immune system, we should always keep in mind that the cells of the innate immune system, just like the macrophages or dendritic cells or natural killer cells, etc., they are equipped with many different types of receptors for recognition of different patterns which are present on pathogens. But these cells, the cells of the adaptive immune system, the B cells and the T cells, they are very specific in identification of pathogens. I told you in my earlier lecture that the B cells has a single, you can say phenotype, a T cell has a single phenotype, one T cell for one type of antigen only, one B cell for one type of antigen only. So what is the capacity of producing, multitude of cells with different receptors? There is a tremendous capacity there. There is reshuffling of these B cells and T cells receptor genes inside your bodies. When these cells are going to mature, then millions and millions of different types of B cells and T cells are produced in your body on daily basis, So if you just compare that number with the number of antigens or pathogens that we know, that is a very small number of pathogens that are infecting human beings. For instance, a couple of hundred of viruses or bacteria or fungi, for instance. But if you look at the diversity of the B cell receptors or the T cell receptors, this is in millions, millions or billions of these different types of receptors are there. It means that the pathogens that already we know about, our immune system already also knows about these different types of pathogens or the moieties that are present on the surface of these pathogens, and the millions and millions of other pathogens that would encounter the human race in future. For each and every of the pathogens, there would be a perfect match in the shape of a B cell receptor or a T cell receptor. So there is no escape from this particular system, which is called the adaptive immune system, because of the diversity of these receptors. If you look here, the B cell receptor actually is an immunoglobulin. Immunoglobulin means that this is an antibody actually, okay? So the B cell is equipped with an antibody, but there is a difference between the antibody and, the B cell receptor. And the difference is only the transmembrane region. The plasma cell is going to secrete these immunoglobulins or antibodies in this form, which would not have a transmembrane region. But the first cell, the first B cell, which is going to encounter a particular antigen, would have this transmembrane region as a B cell receptor. So the B cell receptor essentially is an antibody, but the B cell is not going to secrete that antibody unless the B cell comes in contact with some kind of antigen for which this antibody is specific. It has two domains or two antigen binding sites. You can see here on the right and left side. But if you look at the T cell receptor, it has got only one site for binding of the antigen, as you see the yellow part here. So in this way, you can just compare the binding site of these antigens and how these are going to work in the future. Now, how the antibody is going to work. The B cells would make very specific antibodies for, neutralizing a particular pathogen. So how the antibody is going to do this particular job is going to do this job by two mechanisms. One is the neutralization, and the other is opsonization. Okay, to neutralize the pathogen directly or to enhance its phagocytosis, we call it opsonization. Okay, if you look at this image on the right, there are specific antibodies given in different colors. These are the color codes. There is a yellow antibody, there is a blue antibody, there is a green antibody, three different types of antibodies for raised against three different types of pathogens in the body. So what's going to happen? If there is infection with some bacterial toxins, you see the round balls here on the left, in the left corner, or is infection with some bacteria, in the extracellular space, then how the cells of the innate immune system are going to respond to these different type of cells. If these are the toxins, for instance, produced by bacteria, for each and every toxins there is a specific receptor on the surface of a macrophage or other immune cells. So they are going to bind with their specific receptor. What is the bacteria going to do? The bacteria is going to come in contact with this macrophage, and the macrophage has different strategies. Either it is going to recognize it with different receptors and just phagocytose this, or this process can even be facilitated. Now we are going to discuss neutralization or opsonization and the role of antibody in this particular, these particular processes. That's why if you see here, the red rods, these are bacterial species, and the green antibody has particularly been raised against these red rods. On the other hand, against the toxins, you have the yellow antibody, which already exists there in the body, So, what are the toxins are going to be bound with the yellow antibody. There are specific antibody for the toxins, and the rods are going to be to bind with specific bacterial species. The green with one bacterial species, the blue with another bacterial species. This is just telling you about specificity, that the green is not going to bind to the other pathogen, which is bound with the red, with the blue antibodies. And the blue antibodies are not going to recognize this particular, bacterium on the right side. So for each and every pathogen, there is going to be a specific antibody recognizing that particular pathogen. Okay. So what happens next? Neutralization is that the toxins has been, if you see, bound with these different types of antibodies from all the sides, and now this particular cell, the toxins can normally bind with the cell of the innate immune system, but the antibody also has got receptors on this particular cell of the innate immune system. So all these antibodies now have surrounded the toxins, and they are going to help with speedy internalization of this particular toxins. These toxins would be taken up by the cell. We call it neutralization, or opsonization, on the other hand, to facilitate the phagocytosis of this process of ingestion of these particular bacteria into the cell, where inside the cell, what would happen to them? The cell is going to degrade these pathogens inside. So these are the two processes that can be helped with these antibodies raised against bacteria or their toxins or the viruses that we talk about. One is neutralization, where the antibody is going to bind tightly to a site on the pathogen to prevent growth, replication, or binding to human cell. Okay, so that particular pathogen is not going to bind directly to the human cell because it's already bound with the antibody. Okay, and the binding pattern, which is going to be recognized by the human cell, already is covered or masked with the antibody. Lots of antibodies can attach to the same pathogen and block the entry of, the direct contact of that particular pathogen with the human cell. And opsonization, as the antibody receptors are present on phagocytes, thousands of antibodies bind to pathogens, and the Fc receptors on human cells bind to these antibodies. This is called facilitated phagocytosis, okay? And we also call it opsonization. So this bacterium can be taken up by this particular phagocyte normally, and it would take a bit more time for this particular cell to take up the bacterium. But if there already exists some antibodies against this bacterium in the system, these antibodies can facilitate with this particular process or enhance the speed of this phagocytosis or this process of internalization of the pathogen.